All right, so today we're going to be reading from Modern Biology, and we're going to be reading a portion about Gregor Mendel and Mendelian genetics. So let's get started. In 1843, at the age of 21, Gregor Mendel entered a monastery in Austria. His task of tending the garden gave him time to think and observe the growth of many plants. In 1851, he entered the University of Vienna to study science and math. His mathematics courses included training in the then new field of statistics. Mendel's knowledge of statistics later provided valuable research on heredity, which is the transmission of characteristics from parents to offspring. When Mendel returned to the monastery, he taught in a high school and also kept a garden plot. Although he studied many plants, he's remembered most for his work with species of garden peas. Mendel observed 7 characteristics of pea plants. A characteristic is a heritable feature, such as flower color. Each characteristic occurred in 2 contrasting traits. A trait is a genetically determined variant of a characteristic, such as yellow flower color. The pea characteristics that Mendel observed were plant height, flower position along the stem, pod color, pod appearance, seed texture, seed color, and flower color. Mendel used his knowledge of statistics to analyze his observations of these 7 characteristics. Mendel collected seeds from his pea plants and carefully recorded each plant's traits and seeds. The next year, he planted the seeds. He observed that purple flowering plants grew from most of the seeds obtained from purple flowering plants, but that white flowering plants grew some of the seeds of the purple flowering plants. And when experimenting with the characteristic of plant height, he observed that while tall plants grew from most of the seeds obtained from tall plants, short plants grew from some of the seeds obtained from tall plants. Mendel wanted to find an explanation for such variables. Mendel was able to observe how traits were passed from one generation to the next by carefully controlling how pea plants were pollinated. Pollination occurs when pollen grains produced in the male reproductive parts of a flower, called the anthers, are transferred to the female reproductive parts of the flower, called stigma. Self-pollination occurs when pollen is transferred from the anthers of a flower to the stigma of either that flower or a flower on a different plant. Cross-pollination occurs between flowers of 2 plants. Pea plants normally reproduce through self-pollination. Self-pollination can be prevented by removing all of the anthers from the flower of a plant. Then cross-pollination can be performed by manually transferring pollen from the flower of a second plant to the stigma of the antherless plant. By preventing self-pollination and manually cross-pollinating the pea plants, Mendel selected parent plants that had specific traits and observed the traits that appeared in the offspring. Mendel initially studied each characteristic and its contrasting traits individually. He began by growing plants that were true breeding for each trait. Plants that are true breeding or pure for a trait always produce offspring with that trait when they self-pollinate. For example, pea plants that are true breeding for the trait of yellow pods self-pollinate to produce offspring that have yellow pods. Mendel produced true breeding plants by self-pollinating the pea plants for several generations. He eventually obtained 14 true breeding plant types, one for each of the 14 traits observed. Mendel cross-pollinated pairs of plants that were true breeding for contrasting traits of a single characteristic. He called true breeding parents the pea generation. He cross-pollinated by transferring pollen from the anthers of one plant to the stigma of another plant. For example, if he wanted to cross a plant that was a true breeding for the trait of yellow pods with one that was true breeding for the trait of green pods, he first removed the anthers from the plant that produced green pods. Then he dusted the pollen from a yellow podded plant onto the stigma of a green podded plant and allowed the seeds to develop.